should be the same like as we had last time.
So
there's a request to have the poll in the middle and the presentations on the side.
Yep. Okay.
So where you guys have the poll.
Just take the whole back.
So apparently, maybe run it this way for now and then see how they
Yeah.
go.
Whatever.
Are you guys good for the...
Oh, yeah.
Should be good. Yeah.
Perfect. All right.
Good.
See you guys.
Same one I used last week.
I was trying to get one else in.
I'll hop up after they finish this because I have a feeling that we might have to reboot OBS and this PowerPoint.
Yeah.
I'll see. So hopefully I can just directly go to present this all one, but just in case.
Makes sense. Yeah.
It's good. Are we not recording?
No.
Maybe we're getting better.
More likely because I was actually on time.
Yeah, we're slowly.
Just send it again. I'm just gonna leave this because it's streaming. Streaming live. Yes. You might also want to just tell students that we've been streaming continuously since the beginning of the year. So some people thought we had stopped. It's just I haven't been posting it on purpose.
Live streaming too?
Yeah, but it gets recorded. So
It's all on YouTube, right?
It's all on YouTube, so you can just let people know. It's a good idea would just be to subscribe to the channel or just to do a quick search for it. Yep.
What's it called? HMB300 2019?
We might just go ahead and post them all at once. So some people like the stream, some people don't. And you guys can do whatever you need to. I think it's a live microphone. I'm gonna leave this one here because
It'll capture.
it'll pick up most of what you're saying.
Alright, perfect.
Yeah, feel free whenever you're ready.
Are you ready? Sorry. I'm sitting right there.
By the way, Rena, we got confirmation that we are presenting on Friday at 11 o'clock at MAC. So it's
AM?
Yes, not
Okay.
PM.
Okay. Congrats. Alright.
Yeah,
All good? Do you want to go drop your stuff real quick?
I'm okay. Do they show the things on the side? Where are the notes?
They switched it. Do you want me to get my laptop real quick?
Oh, then I'll get mine.
Yeah, because we don't have a lot of time. I should tell them now then about the YouTube.
Okay.
Hi everybody! Hi! Hope you guys are doing well so far. So before Rena and I start the presentation, I just wanted to let you, Dr. Ju wanted me to let you know that we haven't stopped live streaming all of the classes. So if you are looking for a recording, it's all on a YouTube channel. It's HMB300 2019, I believe. If you look for the first ones, you'll find the channel. It's really easy. So Dr. Ju just stopped posting it on purpose, but it's still all there. We have everything you need. But for today's presentation, we're going to be talking about project management and resources and how to incorporate feedback. This is more applying, we were hoping that this would help most with the written review. Yeah, you're good?
Yep.
Perfect. Yeah, so why want to... How does it go to the next slide? Is it this one or do we go to the PowerPoint?
Let me see. Sorry, it's this one here. And then if you just go...
Perfect. All right, sorry about that. So, can I have your attention, please? Thank you. All right, so there's a lot of resources out here for you that you could use to your benefit. One of the amazing little ones is the UTSC assignment calculator. We will be talking more about that, but that one is a huge, great resource. Another one is the Writing Center. They can help you refine your paper, make it sound right, flow right, all around include setting up appointments, so whether that is with your a learning strategist, your registrar, your writing center, setting those up as soon as you know that you haven't signed a link, they'll actually force you to have something done right before those appointments. And we also want to estimate how long each of those mini chunks will take when you're planning, so you know that you're reasonable with yourself. So if you're still working on the written assignment, finding sources could be one chunk at a time, writing your intro paragraph, your discussion, those could all be separate things, and then finally also make sure to leave room for your citations. And then for those of you who are submitting today, we just thought we could give you some pointers on how to incorporate your feedback. So this sounds super obvious, but we've put down the 3 steps here. So first, you're going to review feedback, understand it, then come up with a plan on how to fix those changes. And I feel like what many of us do, myself included, is when I see feedback, I'm like, oh, okay, I'm just going to fix that quickly. And then what we do is we miss this middle step of trying to understand the feedback, so why we got that. So a comment that you may get on your written assignment is try to clarify this more. So trying to figure out why you're trying to clarify that. Is that because it links to a bigger theme in the course? Is it going to help your assignment flow better? And once you're able to look at it through a more holistic lens, it'll also help you improve future assignments as well. And then another thing that's helpful is when you're writing your assignment, let's say you feel like you were doing really well on one part, you're feeling kind of iffy in another part, but then the comments that you get don't reflect that at all, so you're feeling quite surprised. Take that into account, and that can also help you reevaluate what your priorities were within the assignment. And finally, don't take feedback personally. So, now that we've talked all things academic, we're also going to talk a little bit about fun. So these are some of the upcoming events I found on Facebook. It's always great to, go out to events and have some fun. I know school can be really hard, and those events can be a really nice break for you. It can also really help prevent burnout. We're close to exam season. You don't want to burn yourself out. So yeah, I wish you all good luck for the next few weeks. I believe in you all. Thank you.
Hopefully this works. Yep.
Not sure how well this is going across, but we'll see. All right. All right, so happy Wednesday. Hope your Wednesday's been going okay. I know you have a lot of assignments and you have like lab drafts and stuff due as well. So I hope that you're taking good care of yourself. As Bakiza and Rena had mentioned earlier, it's a really stressful time. You'll know that it's a stressful time because you see all these de-stressor events coming up, and they actually have started very early this year. So I've seen some already beginning this week. So we have about 3 weeks of classes left. So please take good care of yourselves. We have a lot of ground to cover today, and this lecture will also be fairly heavy, so you're going to have to take some notes as indicated on the top left of this particular slide. And if you have any questions, I do have just an open office hour, or if you have like questions about your assignment. Some of you are asking, can you get an extension on your draft? The draft is going to be due today at 8 o'clock, but that doesn't mean that you can't continue to come in and get feedback on your assignments. I'm happy to help you with that. Your TAs are also there to give you suggestions. So don't look at it sort of as a finite, I can't get any more feedback or information or suggestions about my assignment. Look at it rather as we need some time to read through the different things that you're submitting. So please continue to work on it regardless, and then come and see us when you have the opportunity or if you have any other questions. So one of the things that we were looking at in the last lecture is really how do we measure synaptic health? We had learned about this technique using MRS spectroscopy, and this MRS spectroscopy allowed us to look at various molecules, and one in particular is a marker for health. Sorry, I'm just going to grab the laser pointer. This molecule here, N-acetyl aspartate, if your neurons are nice and healthy, you will have a lot of N-acetyl aspartate at the level of the synapse. We can see that on an MRI, and in a particular form of an MRI, known as MRS. And this MRS spectroscopy allows us to see if we have healthy synapses in a living brain, or if we have lower levels, it means that the synapses are not as healthy. And so we looked at a different type of single nucleotide polymorphism around BDNF, and we know that if you have the methionine 66 version of BDNF, that you generally have poorer synapses or unhealthy synapses compared to someone who has valine 66 BDNF isoforms. So this implication means that we can now look at synaptic health in an intact person. We can see if their synapses are developing normally, and it also gives us this opportunity to correlate it or to combine it with data around the genotype. So we can look at a genotype and a phenotype around an important molecule known as BDNF, and it gives us some insight as to whether or not someone is healthy or not. And I did go through in the last lecture some of the different things that are related to MRS spectroscopy. So we look at proton spins, just like a normal MRI, but we're looking at proton spins for specific molecules. Not all molecules, just very, very specific molecules, as I've indicated earlier. And the molecule that is key for healthy synapses is the overall level of N-acetyl aspartate, or NAA. And we normally compare that to a molecule like creatine. So, or creatinine. So when you look at these levels, and if you compare it to creatinine, we will see this peak will go up or it will go down. So this is NAA, N-acetyl aspartate. And for this area of the brain, looking at many, many different synapses, it's probably looking at millions of different synapses at the same time. We are looking at the overall peak of these millions of synapses for this molecule known as NAA. And if I have the methionine 66 version, the overall levels of NAA might be lower. Why is this important? Because we're doing this in comparison to a molecule known as creatinine, which doesn't change. Regardless of the status of your neurons, whether healthy or unhealthy, it is always going to stay the same. It doesn't matter what you're looking at, whether it's valine 66 or methionine 66, the levels of creatinine will always be at this level. And in fact, another related molecule, choline, also has a very sort of consistent signature on an MRS as well. And when we see this doublet on an MRS scan, we know that this next peak is going to be NAA. So this is how we know the molecule that we're looking at. We look at this doublet of choline and creatinine. The next molecule that we should see would be NAA, and we look at the overall size of this peak in comparison to creatinine. Do you have any questions about this, or anything that I can clarify for you now before I go on? Yes. So we're looking at the same region of the brain. So we're just scanning for like a different molecule. Still the same millions of synapses in that area, and that area that we're scanning is in this little red box that you're seeing here. Any other questions that I can answer for you now? No. Yes. Hi. Yes. Yes. So the overall size of the peak, so we wouldn't expect to see a change in the size of the creatinine peak. That'll stay the same regardless of my genotype or the single nucleotide polymorphism I carry. But the NAA levels will either be higher or lower. So it would be lower in an individual with the methionine 66 version, which is associated with poorer memory, might be implicated in other conditions as well. All right, and again, just to re-emphasize this, we are looking at the overall levels of NAA or N-acetyl aspartate versus the creatinine levels. And some areas of the brain are naturally going to have higher levels or higher ratios than others, and so we need to look across a variety of different regions. So again, it's just for your own information, and the key bullet point here is over on the right about the creatinine levels being consistent and being pretty steady regardless genotype within the brain. So one of the practical applications of this, especially if you're in sports or you're an athlete, or it doesn't matter, you could just be walking down the street and developing concussions. Right now, there really isn't a firm diagnostic test for concussion-related injury. And you can think about the NAA levels. Like, so would you expect the NAA, if you've damaged synapses in an area of the brain that you know that has undergone a concussion, would you expect it to change? Well, some people think that it does indeed change, that you can actually see lower levels of NAA, and some people are using this to try to track the progression of things like concussion. And if you look very carefully over here on the y-axis, in fact, we are looking at the overall NAA levels, and we're looking at control individuals versus different times of recovery, and we're looking at these NAA. And some people feel that in addition to diffusion tensor imaging to look at white matter tracks, that NAA levels, or MRS spectroscopy, in other words, is one way to see if someone is getting better. So if your synapses are recovering, the levels of NAA versus creatinine will start to go back up over time. And if you're not doing any better, it may just stay lower. So there are some real-world advantages to this, especially after concussion or after any kind of traumatic brain injury. People are thinking maybe we should be using this to track the progression of synaptic health after different types of psychological injury like depression, etc. So it's a very interesting application. Right now, there's still some controversy about it. Not everyone agrees that this is a good measure of synaptic health in terms of tracking concussions or tracking other things, but I think more and more people are buying on to using this as a potential application for concussions and recovery from concussions. So I'm going to switch gears just a little bit. We're still talking about BDNF, and we're still looking at the overall implications of what I introduced in the last lecture. So in the last lecture, we looked at BDNF having two isoforms. One isoform was good, so valine 66, BDNF, it's good. We get lots of it being secreted. And methionine 66, BDNF, don't get as much of it being secreted. And I sort of mentioned that in passing. But how did scientists actually figure out that that's the key issue? So if you look at where this is in the coding region, a single nucleotide polymorphism shouldn't really make a big impact on the function of this particular BDNF molecule. So scientists wanted to understand this, so they took the two different nucleotide sequences, one that caused a valine at position 66 of the mature BDNF, the other one at methionine 66 of the mature BDNF. So the genetic sequence of this was used in transfection studies in cultured hippocampal neurons. They wanted to introduce these two different BDNF genes into hippocampal neurons. So what happens if I transfect valine 66 isoforms, or what happens if I transfect separately in a different group methionine 66? So it turns out that if you do these transfections on the individual hippocampal neurons that you've transfected into, nothing. So if I have methionine 66 or valine 66, the neurite outgrowth on those cells look identical to each other. If I look at valine 66 or methionine 66, I still produce the same levels of these proteins. So when I take the cells after I transfected them, I do a Western blot, so I purify the protein from culture, and then I run it on a gel. I still see the same levels of BDNF being produced by these particular cells. However, there is a big difference. So valine 66, when you do the immunocytochemistry for this, was found to be in the cell body and in the dendrites, but methionine 66 versions of BDNF was only found around the nucleus. It never really made it out of the nucleus and the Golgi apparatus, in other words, the ER Golgi. It got clustered around the nucleus. So the valine 66, it's out in the dendrites, it's ready to be secreted out, it's ready to be produced and released, versus the methionine 66, I produce exactly the same amount, but I actually can't cause it to be released as easily. And so this was actually a phenomenological description. So they were describing what was happening, but it turns out that you needed to continue this experiment, and I'm going to show you the experiment in the next few slides. So before I get to that part of the experiment, are any of these bullet points ones that I can go back and confirm with you, or if you have any questions to follow up? Do you have any of those that are at the top of your consciousness right now? Anything that I can answer for you? No? So are you conscious, I guess, is the key question. No? Not sure? Not sure is okay. We'll get into that realm of philosophy later on. So the valine 66 is easily secreted. The methionine 66 has very little secretion. You produce it, but it just can't get out of the cell. So again, surprising. So one other thing to keep in mind, you need to be able to relate this back to the in vivo situation. In one of the processes involving LTP, does BDNF help LTP and strengthen synapses or not? If I have BDNF, will I get more LTP or no change? More LTP. So I'm assuming this means it's going to go up, right? Right, awesome. So if it's going to be involved in LTP, LTP requires activity. And it's the same thing here. Scientists wanted to know, how can we show that activity in a neuron will cause more release or it will not change the release? And does the isoform of this actually make a difference? So how do we measure this secretion of BDNF? So here is sort of a diagram. It's a simple diagram. We are using the cytomegalovirus promoter to drive the expression of the BDNF gene. And this BDNF gene is either going to have the nucleotide sequence for producing valine 66 or, in a separate independent experiment done the same way, methionine 66. We want to know that it's being expressed, and we can also use this fusion construct where we have BDNF and GFP. So if I see GFP, does that mean I produce BDNF? Yes. If I produce BDNF, will I see GFP? Right. So if I see green or GFP, BDNF is going to be there. And if I have BDNF, I should also see more green. So keeping that in mind. We also will introduce a separate vector, which is color-coded. So red vectors being delivered, green vectors representing this. And this is just to outline the cell that we are going to express a different gene, which is known as DS-RED or DS-RED2, which is the newer variation. So it's just a fluorescent molecule that the cell, if it takes it up, and only if it takes it up, it will express a red-colored fluorescent protein. Okay? And DS-RED is just a red fluorescent protein. So before I go on with that, is there anything that I can clarify for you now, confirm anything that you're not sure of, or expand on if you need more information? So we're transfecting two different genes, BDNF containing gene and a DS-RED containing gene. So I should see both red and green fluorescence in exactly the same cell. That cell will have both red and green. Are we good so far? If everything works. So here you're seeing a very, very sensitive technique where I can directly inject using an electrode. So we have this large electrode. I can impale the nucleus. I have to look under the microscope, and I can impale the nucleus and directly inject these vectors into the nucleus, and the nucleus will incorporate and then cause the expression of these different proteins. I could also use gene gun biolistics, so I can coat little gold particles with these vectors. So I can take gold, combine it in solution with the DNA, this DNA and this DNA, and then I can directly shoot those gold bullets at the nucleus, and they will penetrate the nucleus and deliver the DNA to the nuclei. So gene gun biolistics is a great way to introduce a gene into slice culture or directly into tissue. We could also do calcium phosphate transfection, and there are multiple ways to use chemicals to deliver the gene into the cell. So transfection, we could use lipofectin, and these often are used in dissociated cell cultures, which means that we have individual cells or we use them in cell lines. Okay, so one method is better for cell slices or tissue, and the other method, transfection, whether we use calcium phosphate transfection or lipofectin-mediated transfection, we are better off using them in cell culture or in cell lines. Yes, there's a hand up at the back. Yes. Okay, so great question. So why do you have two separate vectors? Why don't we have like one vector and introduce both DS-RAD as well as the BDNF/GFP fusion construct, since they're both using CMV? Why not into like one vector? So theoretically, it's possible to do that. You would normally use a vector that is known as a bicistronic or a polycistronic vector, and those are a little bit more challenging to actually introduce into the nucleus and have both of them expressed. Here, we have smaller size plasmids, and the circular DNA is sort of a plasmid vector. They're more easily taken up by cells. The polycistronic ones or bicistronic vectors, where you can have both of them in the same vector, are a little bit more challenging for transfection studies. So that's a great question. Yes? Is the proportion of red and green that you get expressed the same? Not always, but in general, when we are delivering genes in this fashion, we tend to get overexpression. So we get much more than would normally be expected. Of both, correct, yes. BDNF is just to show that that cell also contains BDNF and GFP. You'll understand that reason in a moment. Sorry, the focus of DS-RAD is just to show that that cell has actually taken up both genes. You'll understand that rationale in a moment. Any other questions? Yes. Cytomegalovirus. So oftentimes viruses have these great promoter regions that drive expression of genes. So a virus infects your cells, and the viral promoter region is what we use here, the CMV promoter. So it's actually the promoter region from cytomegalovirus. Okay, any other questions? Ooh. Okay. All right. So here's why we need to understand that. And I guess before we get into that, so if you're still on your poll everywhere, I'm going to try to jump to the next one. So this picture that you see on the screens on the outside, how many neurons do you see? So is it a neuron? Is it a neuron? So I think the clear winner here is that there's one neuron. So for those of you who are brave enough to pick one neuron, are you like 100% sure that it's one neuron? This is like the beauty of being here at U of T. I'm always causing you to doubt yourself, right? Okay, so I'm going to try to address that question. And the answer is, just very unsatisfactorily, it's a little bit of both. It's both 1 as well as 2. Does that make sense? No, of course not. That's okay. I'm going to try to explain that to you in a moment. So I'm going to take you through these different panels, and I'm going to show you how these panels were actually overlaid. This is a technique that some of you will learn if you are doing any kind of cell biology at all. This is known as scanning confocal microscopy. Some of you are using the next generation of it, which is known as a spinning disc laser type of image capture. It doesn't matter what you're looking at. Just remember, everything in this black box, you're not moving the box. You're looking at the same field of view, okay? So just like you're looking at this desk, nothing is going to change on this desk, but what you see or concentrate on, depending on the light that shines on it, maybe the white paper will be more in focus, or maybe you'll see the box of cookies, but the table itself, that black box, is not going to change. So when we're looking at this, I'm going to start from the outside and work my way in, and let me get back to, is it HDMI 3 or 2, or which one is it? Not 1, it's not 3. 3? Sorry, folks, which one, which HDMI? 3? 2? 2? 3, okay. Yep, no, I just need to see it. Okay. So at the top, remember one of the things that we had introduced? We had introduced DS-RED2. Do you remember DS-RED2 from the previous diagram? Yes, okay, so we have DS-RED2. So DS-RED2 is a gene that you are seeing here, and DS-RED2 is, we see red. So did the transfection work? Kind of. Okay, so we see red kind of everywhere. So we see red here, we see this red coming here. So DS-RED is definitely being expressed. Now, remember what was BDNF fused with? GFP, and GFP is green. So do we see green? Who said no? Okay, we do. We see green in this panel. We see green, right? We see green. 100%. We see green. So this is BDNF. We get BDNF. We have DS-RED. Awesome. So then one of the things that we can do is align these two images. We can overlay, and that's what we call it in cell biology. We overlay the images. So we want to know if we combine the two images, do they sit on top of each other, and what does the red and green look like when we overlay the two different images? And when we overlay the images, it doesn't look like they're the same. But I gave you DS-RED2 and BDNF into the same cell, right? So why doesn't it look identical? Yeah, Lauren. BDNF does get localized somewhere, and it was a rhetorical question, by the way, but okay. I'm going to tell you in a moment, but thanks, Lauren. So it does get localized somewhere, so Lauren is correct. So take a look at this. It just looks a little bit weird. So we do get a little bit of green, BDNF. We have DS-RED, and we have more green here in the middle, and we got some co-localization. And whenever we have red and green kind of combining together, which means that they're sitting exactly on the same place, when we add red and green colors together, we get this orange or yellow that says that wherever red is, we also get green. So DS-RED is in the same place as the BDNF. So this might not make a lot of sense to you unless you do the next image, which is I am going to go in now, and I'm going to stain for this other protein that I haven't stained for yet, but I'm going to stain here on the bottom panel in D for this protein known as MAP2. So this is microtubule-associated protein 2. I hope that you remember that from your second-year neurobiology classes. And it's a good marker for neurons. So this is a good neuronal marker, microtubule-associated protein 2. And here, right in the middle, we see that we have this thing that looks like a neuron. In fact, because it is stained with MAP2, MAP2 does not stain glial cells. It does not stain for oligodendrocytes, nor does it stain for microglia. This is a stain only for neurons. I know that we have here in blue, because this is what we're staining for MAP2. MAP2 is stained in blue. We have a neuron smack dab in the middle. Do you all see that neuron? Just say yes. Yes, okay, awesome. By the way, just out of curiosity, because you're going to see this again on the final exam, earlier on when we talked about neurogenesis and then differentiating the cells using fetal bovine serum, so we have these stem cells, and then we have been growing up these neurospheres, and we want to show that we have neurons, oligodendrocytes, and astrocytes all being found from these progenitor cells in the neurosphere. We used a different neuronal marker. Do any of you remember that neuronal marker? Yes, Isis. Sorry? That would be a good marker for earlier immature forms. How about a fully mature neuron? Different marker. Yes. Synapsin would be a good marker for neurons in general, but we used a different one. So you guys are all remembering different parts of it. Does beta-3 tubulin ring a bell at all? No, yes, no, yes. Okay. So I would suggest, and I don't know if you consider it like sort of a challenging question. I kind of look at it as an easier question. If you get a whole bunch of different molecules and you're told that it stains neurons, you should know MAP2 stains neurons and fully mature neurons. Beta-3 tubulin stains fully mature neurons. We have markers like NuN for neurons as well, but they're a little bit earlier in the differentiation process. We've learned about other markers. We haven't really talked about synapsin yet, but here is a marker for these neurons. And does the red overlay perfectly with the blue? No. Well, that's weird. And in fact, I'm going to tell you one other thing. When we look at microtubule-associated protein, and hopefully this will make a little bit of sense to you, because I know you're going to ask this question later in the bottom. So microtubule MAP2 is usually found in the dendrites. So when you're imagining a neuron, where do you see the dendrites? So are they at the end of the axon, or are they part of the cell body? Where do we normally see them? So near the cell body. So this is why this cell body kind of looks that way, and we see these projections coming off that look a lot like dendrites, okay? And that means that this red thing that's coming in is actually a different neuron. I have transfected a neuron that is sitting way out here somewhere. And what we're seeing is this little axon coming in, this little red line that we see coming in here is actually the axon. So we're seeing actually 1.5 cells. So we see this one cell in the middle. Hopefully you all agree there is a cell there. And then we have this other neuron sitting out over here that we have transfected, and we have transfected it with both DS-RED2 as well as with BDNF-GFP, and we are seeing its axon coming in, this long projection from the left-hand side of the picture, and we are seeing the individual synapses that are coming on to this neuron here. Do you all see that? Yes, Bakiza. Right, so great question. So axons are not the same as dendrites. So axons have things like tau associated with them, and they don't have microtubule-associated proteins to cross-link the microtubules. This is not tubulin. So if you're thinking microtubules, this is not, MAP2 is not tubulin. It's the protein that joins the tubulin together. And that protein that joins the tubulins in dendrites is not the same protein that we see in axons. In axons, we use tau, and tau is the protein that joins microtubules. And so the red doesn't have blue because it's an axon. It's not a dendrite. So it's a great question. And this is also why when you're thinking about all those diagrams where the axon looks so much thicker than the dendrites in all of the diagrams, it's completely wrong. The dendrites are so much bigger. The axons are really, really small, which is what you are actually seeing here. So let's go back and think about this picture again. So we have one neuron in the middle. We have this other neuron coming in that has been transfected, is expressing all the genes, and it's making synapses. And I also told you that they don't perfectly overlay with each other. So what we're seeing is that we have a neuron that has both BDNF and DS-RED, and it is releasing the BDNF onto this postsynaptic neuron. So the green that you see peppered on top of the cell at the top, that's the release of BDNF. Yes, Lauren. It is. And it's primarily axonal microtubules. Any other questions that I can answer for you now? So I'm going to, yes, hi. Sorry, how do I know? It looks more like the bottom neuron has, it's being released onto the bottom neuron. Okay, so remember we introduced both red and green? And we did it in a way that is going to make sure that this cell, and only this cell, is going to contain red and green. There's no possibility that I'm going to inject like the cell right beside it. It's only this cell that is doing it, and I'm not doing anything to any other cell in the area. And so when I go in now and I see the release of this green, that could only have come from one cell. And that's why the question earlier, why have that red protein there? I need to know that protein is being expressed from that one cell. So I'm going to take you through this really quickly on this diagram. So this is how we would do it. So we would see a group of neurons, so in cell culture. So you guys have all done cell culture. You guys have all done primary cell culture in your labs, right? So these are what neurons look like in primary culture. So we have one cell body here under phase contrast. We have another one here. You can see some of the dendrites. And you can see another neuron here, some of the dendrites. You might think this is an axon. This is not an axon. Still a dendrite that's associated with this neuron here. And all of these represent cell bodies. Is that good for everyone? Yes, no, maybe? Okay, awesome. So we have all of these different neurons. So I want to use this to explain the different processes. So complementary to what we saw before, I can take an electrode, I can find one cell, the cell up here at the top, and I can inject into the nuclei of this cell these different constructs. I could, in fact, inject in DS red, and if this cell is the only cell that I'm injecting into, it will express DS red and become red. Is that good for everyone? Good? Yes, maybe, no, maybe? Sure, maybe? Okay. And then if I look under fluorescence microscopy, this would be the only cell that I see fluorescing red, right? Nothing else. Nothing else will take up and express that. Good so far? Okay. But that's not the experiment. The experiment is where I introduce two different vectors. So I put both the GFP BDNF and I put the DS red into the same cell. That was the experiment, right? And so if I do the same thing, I told you whenever red and green are co-expressed in this particular neuron, that I would actually get yellow. And so if I'm only looking at this cell and nothing else, and I'm not doing anything else to this cell, then one of the things that I would see is that I would get perfect alignment. Everything on this cell is yellow. Any questions about this so far? Does that make sense to you? So we are listening to the sound of crickets, the crickets. Are we good? We're good, okay. So again, nothing too major there. Here's the thing though. I told you early on, and now you have to kind of put all of this together, and I hope the story has progressed in a logical way that you can put this together, that I have introduced two different genes into this one cell, and the different variants, whether it's methionine 66 or valine 66, one or the other, if I induce activity, if I get this neuron to start being active, I can actually get it to release BDNF. BDNF is coded with green, or it's also expressing GFP. So whenever I see GFP, I also know that BDNF is being released. So when I induce activity in this cell, the green is going to be released. And so I'm not going to get perfect co-registration when I do the overlay. So I'm going to get these little droplets, these little globules, these little fluorescent sort of stains that are being released from this cell here. Is that part okay? Again, activity-dependent release if this is valine 66 BDNF. How many of you are lost? How many of you are lost but don't want to say that you're lost? Blink hard now. Hard blink, now. I can't tell. Okay. So we've introduced two different genes, the red, DS red, it just outlines and fills every part of the cell, and we have BDNF, and the variant of BDNF that we'll be looking at is the valine 66 BDNF. It can get released from a neuron if there is activity, and this is what I'm showing you here. This neuron is the only neuron that we've injected, and the green that you are seeing being released is actually the BDNF that is being released from this cell. So my question is here, and here's where I said that you'll need to take notes. How do I have a group of cells in a dish, and let's assume for the time being I've removed this electrode, so this electrode is actually not there anymore. How do I depolarize or create activity in all of the neurons to cause release? What chemical substance can I add to cause depolarization of neurons? Yes. Sodium. Already a really big driving force for sodium. There's lots of sodium outside the cell. There's a huge driving force. I can look at the Nernst equation. If I double the amount of sodium outside the cell, I'm not going to get more than a small change in the membrane potential. Different idea. I could add a neurotransmitter if I knew what cell type this was, independent of knowing what cell type, to depolarize any neuron. Chlorine. Assume that this is a mature neuron. I feel like I'm doing some kind of legalese. I have to keep going back and making sure that we're following the same path. What one chemical substance? What's a positively charged cation that you are aware of? So sodium's already been mentioned. Potassium. So if I have a lot of potassium outside the cell, I can depolarize the neuron by having a higher potassium outside, which is what was done here. By increasing the amount of potassium, by changing the medium to have more potassium, I induce activity, everything depolarizes, including this cell, and I get release after adding high levels of potassium. Extracellular potassium is a great way to depolarize neurons. So why does it look like that? So it looks like that because we have made synaptic connections to this other neuron, which under fluorescence microscopy actually looks invisible. So I can't see it unless I look under phase contrast microscopy. So I see that there is indeed another neuron, and it's been releasing this BDNF onto this other neuron that we see here. I can also go back and stain for a nuclear dye like Hooks or DAPI to see where the nuclei are, just to get a count of the cells. They didn't do that here, but this is another way that we could do this. And so when we go back to this diagram, I hope that it makes a little bit more sense to you. That in fact, the cell that we've been working with, the one that we injected, is sitting all the way off to the left, and we've been inducing activity, and it's causing the release of BDNF that you are seeing in panel C, and it is releasing this postsynaptically onto the neuron that it's making synaptic connections onto, which is shown in panel F and also shown in panels D and E. Did you have a quick question? In fact, if you look, we're not seeing release of DS red. We're seeing the synapses surrounding the postsynaptic neuron, and the DS red is going through the entire length all the way down to the synapses, and what we see in those little dots are actually the synapses that are surrounding the neuron here in F, and that's why it's not being released. DS red is not being released. The green is, and that's the BDNF. Is there anything else that I can confirm for you, clarify for you? Can I confuse you by adding other things? No. Don't want to do that, but clarify anything for you? Yes, Bakiza?
So we affected the neuron that we can't see, and we can see the axon of? And
Yes. Induced act. Yes. Yep. Any other questions that I can answer for you? So I think now is probably a good time to take a bit of a break. Try to work your way through these images. If you are stuck, there is this paper here at the top. You can go back and read through the review that was published. We'll come back in about 8 minutes to continue on, and we'll see how far we get.
so sad.
I feel you, but that's just. We're out of coffee already?
Are we out of coffee? Is that? Oh no. This is not my day.
Tastes good though. You want to get some milk? What if I like fall asleep because there's no coffee?
Yeah, I know, right?
What will I do? This lecture is so dense.
There's like a couple. Yeah.
Okay, you want one?
Oh, okay. Yeah. How'd your paper?
Is it going? Mine's still going. Yeah. Oh.
I have notes. Which is more than a paper.
But it's not an exam.
My bibliography is stunning. It's beautiful. It's formatted. But that's it. So. You have time. Yes. So, the question we were asking actually was, can no, so it is. So this is what it means. So when we started, the green would have been exactly where we started. However, they were like, potassium is looking up. And so
question. I don't know if you'll actually have the answer.
I probably don't. But
Yeah.
ask away.
So the visual summary, I got the mark back, but I noticed there was no feedback and comments on the assignment. I don't know. I don't have it. Huh. I know you didn't mark it because you didn't email. Anna? Okay.
I'll address that.
Thank you.
All right, everyone. If you're okay with doing so, let's go ahead. Sure, that's fine. No worries. It's okay. All right. So a couple of you are asking for the extra tutorial, so I will be running a tutorial from 4 to 6. It will be in Sid Smith Commons tomorrow. You're free to drop in if you are part of my tutorial group. So I have everyone from T through the Z in my tutorial group. About 20 of you weren't able to get comments back, but everyone else, I think, got comments plus the mark back. I'm not really sure what the issue is there, but I'll look into that if you were in my TA group. But I think the majority of you did get some kind of comments, or if you asked, I went back and made sure that somehow they got uploaded. If you have some concerns, so some of you have indicated that you do, that you'd like someone else to give you comments on it, please talk to your TA first. You can CC me on that message. Talk to your TA first. Ask for some comments if you haven't received them. It might be a technical issue or it might be a technical glitch. Hopefully with some good discussions around your summary, you might get some feedback that makes sense to you or what you could do to improve it using that feedback loop that Bakiza had talked about earlier. So please do so if you can before contacting me. So contacting your TA, because they're the one that looked at it. If you're in my group, you're welcome to ask questions about it. I'm also going to just take any questions that you have. If you have a study group and you just want to join us at Sid Smith Commons tomorrow, you're welcome to do that as well. We'll be there from like 4 to 6 o'clock. You're free to drop in after you meet with your own TAs. But that's the way that I would prefer to go from that, and I've had some questions. So please contact them first, and then come through me if you have additional issues. So I'm going to ask this question because it's an important one. On that previous experiment that we just looked at and we spent some time going over, if you understood what we transfected and you understand that there are two different variants of BDNF, what variant were we using that we saw the release of this green BDNF? It's either valine or methionine 66. Valine. If I did this same experiment again, and I haven't shown it, but if I do the same experiment again, but instead of using valine 66, which is shown in this diagram, I decided to use the single nucleotide polymorphism known as methionine 66, what would you expect to see? Would you, one, expect to see green? Based on what I told you, would the cell that I delivered the GFP BDNF to, would that cell express green? Yes. Would I expect to see, after I added potassium chloride or high potassium outside the cell, would I expect to see green leaving that transfected cell? No. So it stays. It can't get released easily. And that was sort of the bullet point that we started with. In most cases, BDNF can be produced, but if it's not leaving the cell, it's not benefiting its neighbors that are around, and we don't see the benefits of BDNF. And that's why the polymorphism is so important. So this sort of led to a really big race. So once these polymorphisms were identified back in 2003, so Sean Egan showed that BDNF had two different polymorphisms. It's important in learning, memory, in the hippocampus. They showed that it's important for synaptic health. other individuals had also shown around 2001 to 2003, the Kohara paper, that there's a difference in the release of this BDNF isoform. Other people wanted to know what causes or what triggers the release of BDNF, or how can we increase the levels of BDNF within the bloodstream. So this race to try to figure out what these molecules were really started to become a very controversial area. And one of the things that is still under a lot of debate is whether or not this aligns with what I talked about before. So earlier, I told you about neurogenesis. Neurogenesis is known to happen in the adult. And does neurogenesis stay at the same level when I get older? So it's thought to go down or decline as I get older. And some people think it disappears altogether. It's controversial in and of itself. And people wanted to know, like, okay, if BDNF is important, BDNF might play a role in neurogenesis, because we looked at that too, VEGF, BDNF playing a role in neurogenesis. How does all of that get controlled? What controls BDNF that controls neurogenesis, brain health, and everything else? Why does it decline? Why does neurogenesis and synaptic health decline as you get older? And people wanted to know, is there something that makes young people or young animals different than older animals? So this race to kind of look at the effects in the blood of from young animals versus older animals led to a lot of very interesting and controversial findings. And so one of the things that is actually not under dispute, this is not disputed at all. So if you take blood from young animals, you will make the brain of an older animal better. So if you take blood from an animal that is fairly young and you deliver and transfuse that blood into an older animal, the older animal actually gets much better. Its muscle function improves, its brain function improves, its memory improves, and overall the synaptic health, the NAA levels would all start to go back up. So there's something in the blood of younger animals that actually leads to better brain health. So it was really, really fascinating. And in fact, that has translated into people doing this. I don't know if it actually works all that efficiently, but people do this, not so much here in Canada, but in the United States, it is actually sort of an industry of getting blood transfusions from younger, healthy individuals. And we know that this all occurs because of experiments that were done by a number of different groups, but one of these groups is the group from Harvard University by the name of Amy Wagers. And I'll get back to Amy Wagers a little bit later. So Amy Wagers and others did a bunch of experiments to prove that there is indeed something in the blood that causes loss of brain function, decreased neurogenesis, loss of BDNF. All of these things are indeed blood related. So in aging, there is definitely a decrease in neurogenesis, especially in animal models. There is a reduction in synaptic plasticity as you get older, and that means that if you are losing synaptic plasticity, your ability to remember things is lost as you get older. Again, it might be anecdotal, but your parents are probably telling you, my memory isn't as good as it used to be. And there's a biological basis for all of this. If you do something called heterochronic parabiosis, and this is the technique that I'm going to be outlining for you in the next slide. Heterochronic parabiosis is where we can surgically attach the blood vessels from a younger animal to an older animal. And this heterochronic, 2 different ages, parabiosis, the connection of these blood vessels, allows for the young blood to move into the older animal. Is this a hard surgical technique to learn how to do? It's actually one of the easiest surgical techniques that you can learn. So all you have to do is to open up the skin of an older animal and a younger animal and then suture them back together. The blood vessels grow from one animal into the other. And it's pretty simple. So this is fairly easy. And it might be a lot of different things. And we don't know all of the answers yet. And this is why I'm introducing this to you. So we could have some kind of rejuvenation factor going from the younger animal into the older animal. Some of the aging factors might be lost from the older animal and going into the younger animal. There were lots of questions, like how is this working? It's undeniable that it works. So if you do this, the older animal does indeed get better. It rejuvenates. Its function improves quite a lot. We can go back and use BRDU, see increased neurogenesis. Generally, we see an increase in synaptic plasticity, even though here it's an unknown effect. Other people have gone in doing heterochronic parabiosis and shown that we get this much synaptic plasticity in an older animal. After rejuvenation, transferring the blood, we get this amazing increase in synaptic plasticity, and their memory also gets better as well. And this is why this becomes an issue, because this race to find this molecule in the blood from a young animal that transferred over, you can imagine the drug companies would want to find this molecule. They would pay big money to find this molecule. So this race to kind of be the first to discover this molecule in young animals that gets lost as you get older. So as you get older, you get lower and lower levels of this magic molecule that's found in young people or in young blood, was this multi-billion, potentially trillion-dollar race. And this, I think, is why we have some issues in science. And one of the first molecules that came out of a lab out of Harvard University was this factor known as GDF11. And this is a growth-dependent factor 11 that was isolated by Amy Wagers. And she went on to great fame by promoting this molecule. And once it got into like really high impact journals like Cell and other papers, people started to pick apart this molecule. And this is the beginning of sort of our story about why this competition in this field to understand the effective exercise on regulating the release of molecules is so controversial. So again, without going into a lot of detail, this molecule that she had proposed to have been found in the young blood plasma. So it's not the cells, it's really the blood-borne elements, the liquid elements, something found in the liquid portion of the blood, not the red blood cells themselves. So some magic factor that is carried, which is known as GDF11, according to Amy Wagers, was thought to be this important molecule. However, once she started to publish this, Amy Wagers' group actually started to encounter a lot of controversy. When she reported this in Cell, she saw that as animals get older and older, the levels of GDF11 start dropping. This accounts for the loss in neurogenesis. This accounts for loss in synaptic plasticity. And that if she restores GDF11, it would reverse a lot of these age-related effects. And again, it seemed to be like a really great story because it continued on in Science, the same group kind of reporting in slightly different journals. And it seemed, again, really, really good. However, when scientists started to go back and examine some of the research that she had published in both Cell and in Science, saying that GDF11 was the molecule, it gets lower in aging, and if we replace it by injections, we can reverse all of these effects. Other individuals started to play around with this, and it turned out that they could not see the same effects. In fact, the molecule that she had proposed to find that's unique in young blood, that declines and disappears in older blood, looks a lot like a muscle-related factor like myostatin, and that had a completely opposite effect. So this is where our story is now going to begin in terms of exercise. This is the controversy. Identifying molecules in the late 2010s, so 2010, 2011, 12, 13, became the big driving force. If you could find this molecule related to improving your brain, it would be like you would make it. You'd be rich. And that was sort of the drive that really drove much of neuroscience throughout the early 2010s. And Amy Wagers is that one sort of story where we're not really sure if GDF11 is that molecule or not. So going back, now we're going to rethink a little bit about the effects of exercise. So there's one thing about aging and the loss of these factors with aging. Maybe it's GDF11. There's a lot of evidence to now say, no, it's not the right molecule. But if we start going back and now thinking about what can improve like neurogenesis, what can improve things like inflammation that might cause depression, exercise is one of those unicorns that we haven't yet thought about. We've talked about diet, we've talked about other factors, but we haven't really gone into detail about exercise. And we know that exercise primarily involves the contraction of muscles, and these muscles themselves are capable of releasing molecules, which we call myokines, and these myokines can include all sorts of things like VEGF, so VEGF can be released from muscle, insulin growth factor, or insulin-like growth factor 1 can be released from muscle, interleukin 6, and these in turn can remodel the fat cells, so you lose different things from fat cells, and all of these things together, the effect of exercise may eventually somehow cause the increased release of neurotrophic factors like BDNF. So we talked about all the different neurotrophic factors. Every time you contract your muscle, you are going to be releasing from the muscle myokines. So any one of the ones that you see up here would be myokines. And as you remodel the fat, as you start exercising and you remodel fat, your fat will release a number of different molecules as well. Some cytokines, and some cytokines which are not bad, like interleukin 10, which is anti-inflammatory, also gets increasingly released from the adipose tissue as adipokines, as well as another anti-inflammatory molecule from fat cells known as adiponectin. So all of these things are capable of crossing the blood-brain barrier, changing a number of things within the CNS, including things like neurogenesis. It might improve synaptic plasticity. It might increase dendritic modeling, ultimately leading to better memory through hippocampal plasticity, as well as learning and memory. So all of these are good things, and we think that for the most part, this is also not controversial. We know, like there's no doubt, because all of the meta-analyses that you run, the vast majority, like 95% plus, say exercise is beneficial for you. They do all these things. But we don't know how exercise does it. And that's, again, troubling for us as biologists, as neurobiologists. We need to have an understanding of this system to be able to make some good, accurate predictions about what might happen if something gets lost. You might also think that exercise, in theory, could also remodel the gut microbiome, and people have started looking at that as well. And we know that regular exercise could potentially do this through alterations in the gut microbiome. And there are some significant differences in the gut microbiome between lean versus obese individuals. I won't go through this in a lot of detail, but if you look at the clustering of the gut microbiome in lean individuals in red, they're clustered over here. Obese individuals are clustered over here on the left. There is a difference. We can actually see differences in the gut microbiome of individuals that exercise versus ones that don't. So does exercise remodel the gut microbiome? 100%. Do we know what all of this is doing? No. We have like almost no data on how exercise is remodeling it in specific ways. So we don't know. It's a fascinating story, and if you want to look at that in more detail, please do so. You don't need to memorize all of the data on this slide. It's just to show you that yes, it potentially could also work. Exercise could work through gut remodeling of the microbiome, but we're not sure of the mechanisms that underlie this. So let's assume for the moment, because this is the approach that we've been taking, that when we look at health versus disease, and health of the central nervous system versus the diseased nervous system, one of the key factors is inflammation. So we looked at inflammation and depression, and we know that some anti-inflammatories have just as good an effect as antidepressant medication, and exercise potentially could work through an anti-inflammatory mechanism. So if you look at exercise, the key feature of exercise is that it might actually produce an effect that lowers the overall level of inflammation. And because inflammation is so connected to chronic disorders like type 2 diabetes, Inflammation is associated with it. Cardiovascular disease, definitely associated with inflammation. Maybe neurodegenerative diseases, maybe depression. Inflammation might play a role, and exercise may reverse that inflammation and make you better. And many individuals with type 2 diabetes, they're not told to go on different types of meds. They're actually told to go out and exercise more to lower their blood glucose. So it's also been shown for a very long time through unknown mechanisms that exercise has a lot of benefits on mood as well as on your mental health as well as wellness. And there was no biological mechanism, again, until like the early 2010s. In 2013, there were a whole bunch of papers. If you follow like neuroscience for a long time, you can see trends developing. So right now, you're seeing trends developing around the gut microbiome, its impact on different neurotransmitters in the brain. So you're actually seeing those clusters of papers being developed. And in the early 2010s, everyone was seeing these clusters of papers around exercise, molecules, mechanisms, GDF11, and this next molecule that I'll show you in a moment. And in 2013, similar to Amy Wager's, another group came out, and this is Bruce Spiegelman's group, and I'll go through his paper in a moment. They came up with a dramatic hypothesis. We have uncovered the mechanism by which exercise makes your brain better. So every time you exercise, we have linked what happens with the muscle to what happens in your brain. And everyone was like, wow, amazing. And it was really dramatic back in 2013. So here is your muscle. So imagine this being the cell membrane of your muscle. And on your muscle, these cells, your muscle cells, the myocytes or the myofibrils, all of them contain this molecule, which is known as FNDC5. This had been discovered long before 2013, and FNDC5 is an acronym or a short form for fibronectin domain containing protein 5. And what happens is every time you exercise, you get a lot of benefits, but your muscle also increases the level of FNDC5. It increases the production of FNDC5. We get more of this molecule on the cell surface. So FNDC5, as you see on this diagram here, and similar to this bullet point, is a membrane protein. It has a signal peptide on its N-terminal sequence. It has this long extracellular domain, and it is embedded within the membrane through this hydrophobic domain that you see here. It has a very, very short intracellular carboxy tail. And if we look at this sort of molecule, this looks a lot like one of the signaling molecules that you have heard about in physiology. So what does this molecule look like in a physiological context? Signaling molecule, big part of physiology. We're literally half a kilometer away from Toronto's one biomedical Nobel Prize. Do you know that we have a Nobel Prize in biomedicine? So insulin, have you heard of insulin? Do you know Banting Best? Like every lecture, Nobel Prize, Banting Best, Toronto? No? Haven't heard that one recently? Okay. So there was this guy named Banting who was working with Charles Best. They found insulin. Later, it was found that insulin is actually a bigger molecule. It is a prohormone, and it gets cleaved into its active form. And here's this interesting thing. On your muscle cells, muscle releases factors, and this is why we call it a myokine. Muscle releases factors, and one of these factors that it's known to release is known as irisin. And I didn't really cover it at the time because I wanted to cover it now. And irisin is the production of a molecule that contains, for the most part, this fibronectin 3 domain that you see here as it undergoes cleavage of the signal domain, and we also lose this hydrophobic domain. And your muscle has lots of fibronectin, which means your muscle could also produce lots of irisin. Does that make sense to everyone? No, I can't. Okay, I can. So we have this molecule known as FNDC5 found on your muscle. As you exercise, you produce more of this FNDC5, and it can undergo extracellular proteolytic cleavage. And we won't go through all of the different enzymes that can do this, and it cleaves FNDC5 into this molecule known as irisin. So we lose the signal peptide, so it gets cleaved off. We cleave this portion here, and we lose this hydrophobic domain, and this is irisin, this molecule. This molecule then gets released from the muscle into the bloodstream, and irisin in the bloodstream then targets a number of different tissues. One of these tissues is adipose tissue, and it converts white adipose tissue into brown adipose tissue. It changes the mitochondrial proteins to do so, and this brown adipose tissue is, again, thought to be useful in protecting against diabetes, obesity, etc. So this is taking care of that chronic disease of type 2 diabetes. Does not explain the brain yet, but I'm going to get to that in a moment. So as we get to that, and let me take a little bit of a detour. Let's go back to this story about irisin. So this is the story that is developed by Bruce Spiegelman. So strangely, both Amy Wagers as well as Bruce Spiegelman, they're both coming out of Harvard University. There's a lot of pressure for researchers to stay affiliated with these high-profile universities. You can make of that whatever you will, but Bruce Spiegelman is also out of Harvard, and there's a lot of prestige, like submitting a paper coming out of U of T is different than submitting a paper coming out of like Ryerson University or York University. Sorry, it's the truth. So if there's anyone out there from Ryerson or York, you have great research, but U of T, like there's a name brand associated with it, right? So let's be honest. Okay, let's be really honest. So if you could, let's be honest. If you could, would you rather study here or would you rather get a degree from Harvard? Be honest. Why? Yeah, assuming that no issues with tuition, would you rather get a degree from Harvard or U of T? Harvard. Well, it's the same thing when we look at research publications, right? We want to have publications from bigger universities. So he started to look at these mice that were missing. We're not putting down Ryerson nor York University. They're perfectly good universities on their own, but we're just better, that's all. So these knockout mice, research better. Research, not people, not anything else, no. It's just research publications. So knockout mice that were lacking PGC1 alpha, they were hyperactive. They also had lesions in the brain. So they were moving around strangely. They also, when they looked at the brains of these animals, if you knock out PGC1 alpha, they have like really big holes. It kind of looks like Swiss cheese in their brain. So they had significant lesions just by knocking out this PGC1 alpha. So he went into the literature and he started to see that whenever FNDC5 was being expressed, that you also typically saw the expression of PGC1 alpha. So no one had really connected the two. One's muscle, this different factor was being examined more in the brain. What was the link between muscle and brain? Well, research was suggesting that it's also like connected somehow, not just in muscle, but maybe also in brain. There was also some suggestions that maybe both of these factors, both FNDC5 and this molecule that was knocked out in his animals that had holes in their brains actually might play a role in the development of neurons. So he began to develop this hypothesis that FNDC5 and the molecule that is proteolytically cleaved from FNDC5, irisin, this molecule here, irisin, were somehow linked. No one had shown that before. He wanted to show that somehow this PGC1 alpha, which always kind of interacts with FNDC5, he's seeing lesions in the brain when he doesn't have PGC1 alpha, that maybe this FNDC5 or the version of it that gets released from muscle, irisin, might somehow be related to the beneficial effects within the brain. Maybe it can account for a better brain by increasing the levels of BDNF, that factor that we've spent so much time on in the last little while. Maybe this is the factor that also enhances neurogenesis, which we already know happens. If you have BDNF, it enhances neurogenesis. So could he account for all of the good benefits of exercise by somehow linking this molecule FNDC5 that I just showed you on the previous slide with BDNF production? And again, this exercise in BDNF levels, we've already gone through all of the evidence to suggest that it does indeed increase the levels of BDNF, but no one really understood until 2013 how this actually occurred. So he wanted to start looking at this in a little bit more detail back in 2013, and this is the first author on the Spiegelman paper, Jeff Rand, in his 2013 paper. So the mice were put on this 30-day endurance training regimen, and they had access to a running wheel, and they were running the equivalent of about 5K each and every night. It sounds like a lot, and it sounds like it's actually bad for the mice, but they actually love running on a wheel. So if you put a wheel in their cage, they will go ahead and use it. They'll run. And it's not like they're being forced to run. They just love doing it, so they'll go out and exercise. And they're training on average about 5,000 or 5K every night. So mice that engaged in regular exercise, that had a running wheel, in other words, in their cage, had much more of this FNDC5 in their hippocampus. And he was able to show that this is not in muscle, this is now in the brain. So in muscle, we know that FNDC5 production increases. He's now showing this in the brain. So this might account for the increased learning, which he was definitely able to also show. So mice that exercise more had better learning, learned quicker, had better memory. He was also able to show there was more neurogenesis. And so he thought that this was linked. More FNDC5 in the brain, in the hippocampus, due to exercise, meant that somehow these were all linked together. There had to be a co-activator, though. So this PGC1 alpha also had to be present in order for this FNDC5 production to be increased. So that's sort of where he was sitting at. He developed this model system that I'm running through here with you. So again, I'm going to go through some of the steps in the next few slides, and some of it will make sense if you go back and revisit it. So physical activity, contracting skeletal muscle, and it's skeletal striated muscle, so we don't really see a lot of this in your heart. We don't see a lot of this within like smooth muscle. So skeletal striated muscle has a lot of this physical activity, will cause an upregulation of PGC1 alpha, that co-activator. And this co-activation of PGC1 alpha that he found was essential would cause an increased production of FNDC5. We're still looking at muscle, by the way. We know that FNDC5 can be proteolytically cleaved, go back a few slides, to produce irisin. And his hypothesis was to link what he saw in the brain, PGC1 alpha, FNDC5 in the brain, how does exercise impact the brain directly, that somehow this irisin must make its way back into the nervous system to cause an increase in neurogenesis, etc. We also know that there's a lot of other things that irisin does, and these parts are not controversial at all. So I'm just mentioning that outright. This arm, how it works on the nervous system, still up for a lot of debate. There is no debate that irisin can remodel mitochondria, and it will convert white adipose tissue into brown adipose tissue or BAT cells, and it will help to cause an increased heat production from brown adipose tissue, so you get better energy expenditure, you increase insulin sensitivity, you burn off calories through heat production more effectively, and all of this is taking place because irisin is remodeling the brown adipose tissue. And we also know that in the mitochondria directly, that we are decreasing the activation of white adipose tissue genes, the genes that allow for your fat cells to accumulate more lipids. Those are going down. Uncoupling protein 1 levels are going up. The mitochondria increase oxygen consumption in the presence of irisin, and that's all important. How this works in the brain, though, I'm going to tell you, is still controversial in 2019. We are not sure anymore. These other pathways down here, we are 100% sure that these work. But this pathway that I'm going to show you next is up for debate. So here is his theory. that exercise, and this is also changed, by the way, in 2019. I'll put up that paper at the beginning of the next lecture. So up until last year, this was valid. This is no longer valid, but I'll still go through it. Do you need to exercise more than once to get benefit on mood, benefit on your brain, and everything else? How many of you say yes? Okay, yes? Majority of you say yes? Or do you have to exercise more than once? Continued exercise. So if I go out there today, and I can't do this, but imagine I could run like 1 kilometer. I might be able to run 100 meters, maybe. So if I do that, will I benefit? Will my brain benefit? Yes. Yes, 100% sure? And I never exercise again. Will it be good for my brain? You're all saying no. But you were right the first time. There's one paper that came out earlier this year suggesting that one bout of exercise, just one, is enough to make your brain better. It's controversial in and of itself. So here's why we think that one bout of exercise in 2019, last year, 2018, was not good. So exercise, one bout of exercise, and this is a muscle cell that we're seeing here. This is one skeletal muscle cell. Exercise causes the contraction of muscle. It increases intracellular levels of calcium, which will then activate in the muscle cell, striated muscle, calcineurin as well as CAM kinases. This in turn will activate CREB, cyclic AMP response element B, NFAT, MEF2C, and MEF2D. And these factors, any one of them, so any one of CREB or NFAT or MF2C or MF2D, will cause the increased transcription of PGC1 alpha. Okay, so are you with me so far? This is one-time exercise. If I went out and tried running 90 meters or 80 meters or 10 meters, maybe, on a good day. So this leads to the increased production of PGC1 alpha in the cytoplasm. Right? So I produce more PGC1 alpha. But then I stopped exercising. That was it. So I stopped going out to dragon boat. I stopped practicing. I stopped exercising. What happens to that PGC1 alpha that I've just built up? Well, it gets degraded by the proteasome. So that PGC1 alpha gets lost with time. Anecdotally, that would make a lot of sense. And in 2018, this is what made sense. In 2019, it's again confused. But this is where I think most of you are thinking about the beneficial effects of exercise. You have to continue doing it, prolonged exercise, not just once, but you have to hit the gym a couple of times a week, right? I'm thinking, most of you are thinking, yeah, that's what I mean by exercise. By the way, does this say that it has to be aerobic or anaerobic? And again, also controversial. So can you get all the benefits of exercise through aerobic exercise? Yes. Can you also get some benefit just by lifting weights or resistance training? Also, yes. Again, very, very controversial. So let's assume that you've done one bout of exercise. You did this earlier in the week. You did this on Monday. Today it's Wednesday, so you've been sitting in lecture from 4 to 6. You want to hit the gym. And since Monday, your PGC1 alpha has sort of just been sitting here. Now you go out and do this exercise again, so you've done it more than once. And perhaps a better word than prolonged exercise might be repeated exercise. So it doesn't matter the duration. 30 minutes is enough to initiate this pathway. And so we increase calcium. And again, we will now stimulate a slightly different pathway. We still do this, by the way. This pathway is still active. If you're thinking, no, we only do this, not true. Now we have increased levels of calcium, and we will also be able to stimulate AMP kinase. And one of the reasons why this pathway is important is this AMP kinase will be able to phosphorylate any pre-existing PGC1 alpha. Transcription and translation take time to occur, but if you have any PGC1 alpha left over from the previous exercise, it phosphorylates it. Now, why is this important? Well, this is important because now PGC1 alpha, remember Bruce Spiegelman said it was a co-activator. You needed both PGC1 alpha and FNDC5. Here's why you need PGC1 alpha to make FNDC5. Earlier I told you the more you exercise, the more FNDC5 you make. It's because of this PGC1 alpha and the phosphorylated version of PGC1 alpha that Bruce Spiegelman thinks works as a transcriptional factor to increase the production of FNDC5, which does happen. And this will also cause more expression on the cell surface. It gets cleaved, and we get more irisin formation. So, is this part clear for all of you? Is there anything that I can confirm, clarify, or to go back and repeat for you at all? So ultimately, with prolonged exercise, I make more FNDC5 through this PGC1 alpha pathway. I will also be able to develop more irisin. I haven't talked about irisin, but I showed you on an earlier diagram, irisin is one of the myokines released from muscle factors. Okay, and now we're going to get into what that means. Yes? So some labs have confirmed that it is indeed a transcriptional activator, and the phosphorylated version in particular is a transcriptional activator. Yes. All right. So this is Bruce Spiegelman, and his idea is that somehow in skeletal muscle and in the striated muscle, these pathways that I just mentioned to you, the production of PGC1 alpha binding to the regulatory promoter domain of FNDC5 will produce more of this FNDC5. More FNDC5, after it gets proteolytically cleaved, can form more of this irisin. And this irisin can then enter into the bloodstream and then interact with the irisin receptor and convert white adipocytes or white adipose tissue into brown adipose tissue or brown adipocytes, causing more mitochondrial activation, uncoupling protein, etc. And he feels that this can get into the brain and at the level of the brain interact with some kind of extracellular factor receptor. He didn't call it the irisin receptor, that can then cause, through an unknown mechanism, transcriptional regulation to increase the production in the brain of BDNF. But he did show that it's irisin dependent and that we need irisin to cause increased BDNF production in the brain. So by exercising, he was able to link all of these separate pieces that had been shown in the literature and was able to show that it did indeed cause the production of BDNF. And this was a story in 2013 that finally provided a molecular mechanism that linked exercise and muscle contraction with the improvements in the brain through that BDNF factor that we've always been talking about. BDNF is good for the brain, and how do we make more of this BDNF through exercise? This is the way that he thinks, or he showed, that all of this occurred. So this irisin FNDC signaling pathway that I just showed you on that previous diagram was thought to be really, really important in how the brain gets regulated. And this, again, is the original article that you can go back and take a look at. And apologies, I gave you the name Jeff Ran, it's Christiane Ran, and this is Bruce Spiegelman's paper. But it does potentially give you mechanisms for improving things like major depression. It might be good for neurodegeneration. We don't know, and there's a lot of hypotheses that were generated at the time of the publication, if perhaps meditation, which has also been shown to be impactful for the brain, or simply sleep will alter the levels of irisin. Just being able to meditate in a different way through prayer might also be thought to be important. And we'll get to this in the very last sets of lectures about the mind and body link. And then what regulates all of this? These were big questions, but people were so excited that they had something to work with, the framework to start to build on all of these different research questions. And that's why research is important, because research generates more questions that you can then go out and start answering. The problem is, after his publication, even more so than with Amy Wager's paper, so Amy Wager's paper was attacked like viciously. It was like bad. His was worse. Like, people came out and basically called him a liar, and they were publishing as much as they possibly could as soon as this paper came out. And when you start seeing articles that actually say that it's a myth, that it doesn't exist, you haven't found anything, irisin actually doesn't make it to the brain, we can't find it, show us irisin, would you be worried? Yes. I would be worried, because if you get all of these papers coming out, in great detail, by the way. And it's not just one group, like many groups came out at the same time. We can't reproduce your results. We don't know where iRisin is. We can't detect iRisin. And iRisin having many, many issues. People were like, I don't know what's kind of happening. Should I believe your research or not? So if you're Bruce Spiegelman, what do you do? Attack back, or cry and then attack back. I'm not sure, but Bruce Spiegelman actually came back really, really quickly. This actually, so I want you to understand the context of this. So he defended himself like much more so than Amy Wagers did. Amy Wagers, had different sorts of quotes and other things, but he came back with like a series of publications just really attacking the integrity of the scientists who are publishing that iRisin is a myth, and it's not a myth. So once you start name calling like it's a myth, it's not a myth. It's not a really good scenario to be in. And I wanted to just highlight this, and then I'll come back next week and actually go through the data. This is going to be an important lecture too, and this will be the last of the exercise lectures. And I wanted to just point this out. So he started getting attacked. He published in 2013. He started getting, the paper got out there. People started reproducing his results in 2014. Can't do it. 2015 rolls around. All these papers come out saying, nope, sorry. Like 2 years later, sorry, we can't reproduce your results. It's all a myth. You're basically a liar, but we're not going to call you a liar. We're going to call you a myther or something. And then he's coming out now, just literally months later, April to July. He's coming out. Again, I don't know how much you know about publications, but he had submitted this paper to a high-ranking journal like Cell. It was received by Cell in July of 2015. It was revised. It was asked for some revisions, so about 27 days later, it was revised, resubmitted to Cell, and then it was accepted 1 week later, and then it was published. There is no way that you can publish a Cell impact-type journal that quickly. Like, literally, this is like about a month and a half to get a Cell paper. Unheard of, unheard of. And it's only because of the controversy that existed around iRisin and the stakes. $1 trillion industry. Novartis had invested all of this money in believing that iRisin was the molecule, and they're now like knocking on his door. Hey, Bruce, those papers that you published, are they right or are they wrong? That $1 trillion, we'd like it back. So it's a big sort of question, and he addressed it in a very elegant way. I'm going to go through this paper with you in the next lecture. Again, notice that it's still Christian Rand, so we get the same authors. Bruce Spiegelman is still there. And I want us to think about science critically. I'm going to take you through his data. You need to know a little bit about protein and about protein translation, but we'll go through that in the next lecture. In the interim, just as a reminder, tomorrow, we will have like extra office hours. Please drop by if you have questions or just to hang out. Friday, we're going to try to have a de-stressor over at New College. You're welcome to join that as well. And starting in about 2 weeks, on Saturdays, I will also have office hours on campus as well. So be on the lookout for those announcements. There is a quiz this week, so please make sure that you're staying on top of that. And have a good week. I'll see you all next Wednesday.
So it looks kind of terrible right now because I'm just planning to not cut it until after Christmas. So I decided to suffer the 2 weeks now to save me trouble later. What? This? No. This? Also no. I don't know what happened to the bag actually. Currently in use. Oh, because the power pump's open. Does this mouse work? Oh no. I hate the trackpad so much. Oh, I didn't stop the recording. Oh my god. They're gonna hear me talking. Ah! Okay. Oh.